Buried inside every human genome, making up roughly 8% of your entire DNA, sits a strange collection of genetic fossils, remnants of ancient viruses that infected our distant ancestors millions of years ago and somehow got permanently written into our genetic code. For decades, scientists mostly treated this genetic material as leftover junk, harmless debris from ancient infections that had long since lost any real function. Then, in 2024, a team of researchers made a discovery that changed everything, finding that these ancient viral remnants are not junk at all. They are quietly ticking away like a hidden biological clock. And the specific way they behave can predict how fast a person is actually aging with remarkable precision. This is the real story behind claims that scientists found a hidden code in ancient DNA controlling how fast humans age. And unlike a lot of viral science headlines, this one holds up. To understand what researchers actually found, you first need to understand a little about where this ancient DNA came from in the first place. Over the course of human evolutionary history, spanning tens of millions of years, our ancestors were repeatedly infected by viruses that worked their way into reproductive cells and got copied directly into the genetic code passed down to every subsequent generation. These viral fragments, called retroelements, accumulated generation after generation until they became a permanent, inherited part of the human genome, no longer capable of causing active infection, but still sitting there, embedded in our DNA, outnumbering our actual functional genes by a significant margin. For most of the history of genetics research, these retroelements were considered a curiosity at best, an evolutionary leftover with no meaningful role in how the body actually functions on a day-to-day basis. The 2024 discovery, published in the scientific journal Aging Cell by a research team including scientists Lechaumois and Lovu and Matthew Corley, flipped this assumption on its head. The researchers focused specifically on a chemical process called DNA, methylation, which involves small molecular tags attaching themselves to specific locations along a strand of DNA, effectively acting like a dimmer switch that can turn a particular stretch of genetic code up or down without actually changing the underlying genetic sequence itself. Scientists had already known for years that methylation patterns across the broader genome tend to shift in predictable ways as a person ages, and this earlier work led to the creation of what researchers call epigenetic clocks, mathematical models capable of estimating a person's biological age purely by analyzing their methylation patterns. What made the 2024 study different is that instead of looking at methylation patterns across ordinary functional genes, the research team focused specifically on the ancient viral retroelements scattered throughout the genome, and discovered that these normally dormant genetic fossils show their own distinct, highly precise methylation signature that tracks biological aging in a way that does not overlap with any previously known epigenetic clock. This new tool, which researchers named the retroelement clock, or retroage for short, was built by analyzing blood and tissue samples collected from more than 12,000 people spanning a wide range of ages, allowing scientists to identify exactly which methylation changes within these ancient viral remnants correlate most reliably with biological aging. What makes this discovery particularly significant is that it represents an entirely new biological signal, distinct from the mechanisms captured by earlier, more famous epigenetic clocks that focused on ordinary functional genes. In other words, scientists were not simply refining an existing measurement, they uncovered an entirely separate biological pathway hiding inside DNA that most researchers had written off as irrelevant leftover genetic material decades ago. The technical process behind building a clock like this involves considerably more work than simply eyeballing a handful of genetic locations and noticing they change with age. Researchers first needed to catalog which specific retroelements across the genome show any measurable change in methylation at all, a task complicated by the sheer scale of these ancient viral sequences, since retroelements make up such a large proportion of total human DNA that identifying which specific locations carry a genuinely useful aging signal, rather than simply random biological noise, required extensive statistical filtering across their entire 12,000-person sample set. Once candidate locations were identified, the team then had to confirm that the methylation patterns at these retroelement sites tracked biological age, independently of the signals already captured by earlier clocks like Horvath's original model, essentially proving that retroage was measuring something genuinely new, rather than simply rediscovering a pattern scientists already knew about through a different technical lens. This kind of rigorous cross-validation is exactly why this discovery took researchers years of careful data collection and analysis, rather than emerging as a single, sudden breakthrough. To fully appreciate why this discovery matters, it helps to understand the broader history of epigenetic aging research that came before it. The very first epigenetic clock was developed back in 2013 by a UCLA researcher named Steve Horvath. who spent years analyzing methylation data before identifying 353 specific locations across the genome, known as CPG sites, where methylation patterns change in a remarkably consistent, predictable way as people age. Horvath's clock could estimate a person's biological age using nothing more than a blood, saliva, or tissue sample. And remarkably, it worked with similar accuracy regardless of which tissue type was analyzed, suggesting some kind of fundamental body-wide aging process was being captured by these particular genetic markers. This breakthrough essentially proved for the first time that biological age, meaning how old your cells and tissues actually behave, is not necessarily the same number as your chronological age, meaning simply how many years have passed since you were born. There's also a compelling biological explanation for why retroelements specifically might serve as such a precise aging signal, one connected to how cells actively work to keep these ancient viral remnants suppressed throughout a person's life. Because retroelements originally came from actual viruses capable of copying themselves and inserting new copies throughout the genome, cells have evolved dedicated molecular machinery whose entire job is to keep these sequences tightly locked down and prevented from becoming active again, since an unsuppressed retroelement can potentially still copy itself into new locations in the genome, causing genuine damage if left unchecked. Maintaining this suppression requires continuous cellular energy and precise molecular regulation, and researchers studying the new retroelement clock suspect that as cells age, this ongoing suppression system gradually becomes less efficient, allowing subtle shifts in methylation at these ancient viral locations to occur in ways that reliably track how well a person's cellular maintenance systems are holding up overall. Beyond the purely scientific fascination of this discovery, there's also a growing practical dimension to epigenetic aging research that extends directly into everyday health decisions people can actually make. Multiple studies analyzing epigenetic clock data alongside lifestyle information have found measurable connections between specific daily habits and how quickly a person's biological age advances relative to their actual chronological age. Research following participants on a Mediterranean-style diet rich in vegetables, fish, and healthy fats found their biological age, measured through epigenetic clock analysis, came out roughly a year and a half younger on average, compared to participants whose diets relied more heavily on processed foods, animal fats, and added sugar. Regular exercise and sufficient high-quality sleep have also been repeatedly linked to slower epigenetic aging across multiple independent studies, giving these clocks a genuinely useful role beyond pure scientific curiosity, since they offer people a way to actually measure whether specific lifestyle changes are producing a real detectable biological effect, rather than simply feeling healthier in a vague, subjective sense. The practical applications of this research extend well beyond individual health tracking as well, and pharmaceutical companies along with academic research labs around the world are now using epigenetic clocks, including newer tools built on this retroelement discovery, to test whether experimental anti-aging treatments and cellular rejuvenation therapies are actually working at the molecular level. Before these clocks existed, researchers testing a potential anti-aging intervention had very few reliable ways to measure success beyond simply waiting years or decades to see whether treated subjects lived measurably longer than untreated ones, an approach that made rapid scientific progress in this field extraordinarily slow and expensive. Epigenetic clocks changed that equation entirely, giving researchers a way to test whether a treatment shifts a subject's biological age measurably within weeks or months, rather than requiring an entire additional lifespan of observation. And this has directly accelerated the pace of experimental anti-aging research across the entire field over just the past several years. Some of the most promising recent experiments in this space involve attempts to partially reverse epigenetic age, rather than simply slowing its progression. And researchers at Stanford University have published early results suggesting that certain reprogramming techniques capable of resetting specific epigenetic markers can restore measurably younger function in aged tissue, including preliminary work showing partial restoration of vision in aged laboratory animals through targeted epigenetic reprogramming. These experiments remain firmly in the research stage, rather than anything close to a proven medical treatment available to the public, and researchers involved in this work are careful to emphasize how much remains unknown about the long-term safety and broader effects of deliberately manipulating these epigenetic markers in living tissue. Still, the mere existence of experiments capable of producing measurable, clock-verified reversals in biological age, even in limited laboratory settings, represents a significant shift from where this entire field stood just a decade ago, when epigenetic clocks existed purely as a way to observe and measure aging, rather than any serious attempt to actively intervene in the process itself. If you find this kind of deep dive into the hidden machinery buried inside your own DNA fascinating, go ahead and hit that subscribe button, because there is a lot more of this kind of cutting-edge biology still to explore. It is worth noting, though, that scientists working in this field remain appropriately cautious about exactly what these clocks are measuring at the deepest mechanistic level. Steve Horvath himself has pointed out that if any single genetic marker used in these clocks played a direct causal role in actually killing cells or shortening lifespan, that marker would likely become rarer among older individuals precisely because it would be actively removing them from the surviving population, which paradoxically makes it less likely such a marker would ever have been selected as a reliable clock component in the first place. This subtlety means researchers still describe epigenetic clocks, including the new retroelement-based version, as capturing an emergent signal correlated with the underlying aging process, rather than definitively proving they have identified the actual root cause of aging itself. Despite these remaining scientific uncertainties, the practical value of this research is already becoming difficult to ignore. What began in 2013 with Horvath's original 353 genetic markers has expanded, just over a decade later, into an entire toolkit of increasingly sophisticated biological clocks, each one capturing a slightly different dimension of how the human body actually ages at the molecular level. The 2024 discovery hiding inside ancient viral DNA represents perhaps the most unexpected addition to that toolkit yet, transforming genetic material that scientists spent decades dismissing as evolutionary junk into one of the most promising new frontiers in aging research available today. It stands as a genuinely striking reminder that even the parts of our own DNA long assumed to be meaningless leftovers from our evolutionary past may still be quietly running a program that shapes exactly how and how quickly each one of us grows old. Looking ahead, researchers involved in the retroelement clock project have indicated this is very likely just the beginning of a much larger investigation into how ancient viral DNA interacts with the aging process across different tissues, organs, and disease states throughout the body. Early follow-up work is already underway, examining whether retroage scores differ meaningfully between healthy individuals and those diagnosed with specific age-related conditions, including certain cancers and neurodegenerative diseases, since any consistent difference could eventually point researchers toward entirely new therapeutic targets nobody had previously considered relevant to these conditions. Given how recently this particular clock was developed, much of this downstream research remains in its earliest stages. But the scientific community's response to the initial 2024 findings has been notably enthusiastic, with multiple independent laboratories already working to replicate and extend the original results using their own patient datasets. If this pattern holds, ancient viral DNA, once considered nothing more than evolutionary debris, may end up occupying a central position in how scientists understand, measure, and one day potentially even influence the human aging process for years to come. This theory dovetails with a broader shift happening across the field of aging biology more generally, where researchers increasingly view aging not as a single unified process with one root cause, but as the combined result of multiple separate, only loosely related biological systems gradually losing efficiency at different rates within the same body. Under this more modern view, a person's overall aging trajectory might be shaped simultaneously by how well their mitochondria continue producing cellular energy, how effectively their immune system clears out damaged or malfunctioning cells, how efficiently their body repairs everyday DNA damage, and now, according to this newest research, how well their cells continue suppressing ancient viral genetic material that has been sitting dormant in the genome for millions of years. Retroage offers researchers a direct window into this specific, previously invisible piece of the aging puzzle, one that appears to operate largely independently from the biological processes captured by earlier clocks like Horvath's original model, or the mortality-focused GrimAge. Since Horvath's original discovery, the field of epigenetic clocks has grown considerably more sophisticated, with researchers developing increasingly specialized tools designed to capture different dimensions of the aging process. A clock called GrimAge, developed a few years after Horvath's original model, combines methylation data with additional biological markers to predict a person's overall mortality risk with impressive accuracy, becoming one of the strongest available predictors of long-term health outcomes based purely on a DNA sample. Another tool called Dunedin Pace takes a slightly different approach entirely, measuring not just where a person currently sits on the biological aging scale, but how quickly they are currently moving along it, effectively capturing the pace of aging happening in real time rather than a single static snapshot. Each of these tools works by identifying subtle cumulative chemical changes written directly into a person's DNA, changes that reflect not just the simple passage of time, but the accumulated biological impact of stress, inflammation, diet, sleep, and countless other factors a person experiences throughout their life. What the 2024 retroelement discovery adds to this picture is a genuinely new dimension that none of the earlier clocks had captured. Because retroage focuses specifically on ancient viral DNA rather than ordinary functional genes, researchers believe it may be picking up on a distinct biological process related to how well a cell maintains control over these normally silenced genetic elements as it ages. Younger, healthier cells appear to keep these ancient viral remnants tightly suppressed, essentially locked away and prevented from causing any disruption, while aging cells gradually lose some of that tight regulatory control, allowing the retroelements to become subtly more active in ways that current epigenetic clocks based on ordinary genes simply do not detect. This loss of control over ancient viral DNA as cells age fits into a broader theory some researchers have proposed, suggesting that one of the underlying drivers of biological aging itself may be the body's slowly weakening ability to keep these ancient genetic remnants properly silenced over time. This idea connects to a fascinating and still actively debated question at the very center of aging research, namely what actually causes aging at the molecular level in the first place. For a long time, the dominant explanation among scientists was relatively straightforward, suggesting that aging results primarily from the gradual accumulation of direct mutations and physical damage to the DNA itself, similar to how a car slowly wears out from the accumulated stress of everyday use over many years. This explanation makes intuitive sense, but has always struggled to fully account for certain observations, including the existence of animals and even individual humans who show remarkably little physical or functional decline despite carrying the same accumulated genetic damage as their more rapidly aging peers. This gap in the classic mutation-based theory of aging is part of what pushed researchers toward exploring epigenetics instead. The idea that aging might be driven less by permanent damage to the genetic code itself, and more by gradual, potentially reversible changes in how that code gets read, expressed, and regulated over time. The epigenetic theory of aging, first proposed in a more unified form around 2010 and later refined significantly by Horvath and his collaborators, suggests that biological aging emerges as an unintended side effect of the very same molecular systems responsible for normal development and ongoing tissue maintenance throughout life. Under this framework, the same regulatory machinery that carefully controls which genes turn on and off during childhood growth and development continues, operating throughout adulthood, and the gradual drift and accumulated imprecision in that regulatory system over time is what shows up as the predictable methylation changes captured by epigenetic clocks. If this theory holds up under continued research, it would mean the ancient viral retroelements identified in the 2024 study are not just a passive biomarker researchers happen to notice. They may represent a genuinely important piece of the actual biological machinery responsible for aging itself, rather than simply a convenient genetic feature that correlates with age for reasons unrelated to the underlying aging process. One particularly interesting and slightly counterintuitive finding to emerge from this broader field of research involves the relationship between the specific genetic locations that change with age and the locations that actually determine how long a species is capable of living in the first place. Researchers studying epigenetic patterns across multiple different mammal species discovered that the genetic locations which reliably track chronological age within an individual are often completely different from the genetic locations that determine the maximum possible lifespan for that particular species as a whole. In other words, the part of the genome that tells you how old a specific mouse or human currently is turns out to be largely separate from the part of the genome that determines whether that species is capable of living 2 years or 200 years in the first place, a distinction researchers describe as genuinely surprising and still not fully understood. This finding takes on additional significance when considered alongside the retroelement discovery, since researchers comparing methylation patterns across species with very different natural lifespans have noticed intriguing differences in how tightly various species manage to keep their own ancient viral remnants suppressed over the course of their lives. Species known for exceptionally long lifespans relative to their body size, including certain whales, tortoises, and specific rodent species studied extensively in longevity research, appear to maintain considerably tighter control over their retroelements well into old age compared to shorter-lived species, raising the tantalizing possibility that an animal's ability to keep this ancient viral genetic material properly suppressed may itself be connected to how slowly or quickly that species tends to age overall. While this comparative research remains in relatively early stages, it represents exactly the kind of cross-species investigation that evolutionary biologists hope will eventually reveal not just how aging can be measured, but why different species age at such dramatically different rates in the first place, despite sharing the overwhelming majority of their genetic code with one another.